Okay, so this is the second video, and we're going to look at those coordinates that we just saw, and we're going to explore them in the Ensembl browser. I'll just take a moment to introduce you to the Ensembl homepage and point out some of the important things that are there. So one of the major things is this blue bar along the top. This blue bar is visible in every page that you go to in Ensembl. It has a link back to home, it has a search box, and it also has things like help and documentation. We also have some tools that you might want to use here. We have some news, what's come out in the latest release of Ensembl. We have some genomes here, so you can see what genomes we have available. So you can go from here, you can go into a full list. And so you can see everything that's there with the common and the scientific names, and you can filter the table to find your genome of interest. So if I type in human, I will find human here. So if I click on this, you'll see I have some example data that I can go to. And I also have this more information and statistics. If I go here, I can see information about how the genome was produced, how the gene annotation was done, and I can also see some statistics about the genome. So things like number of genes, size of genome. I'm just going to click on this link here. This has this other assemblies link here. I just want to show you the GRCH37 site. So if you are working with the older version of the genome, you may wish to go to the GRCH37 site, which you'll see is exactly the same. It's different coloured at the top, but it's the same site as we were using before. I'll just click back on this link to the main site, and I'm going to search for this region in humans. I'm going to paste in the coordinates that we were looking at in the presentation earlier. So it's human space chromosome colon start dash end, and I'm going to hit go. The alternative, instead of putting human here, I could have selected human from this drop down instead. I'm going to hit go, and it takes me to my region of interest. So the region, one thing to point out that you will see all over Ensembl is these little question mark icons. If you're ever not sure what you're looking at, click on the question mark and it opens this help page for you. So it gives you a diagram of the page, which is labelled. This is quite a complicated page, so it's got a really long help page. Some of them are a lot shorter than this. There's lots and lots of stuff, and in case you haven't had enough of videos, what you'll see in a moment is Denise, who used to work for our team, giving you an intro.
Hello, I'm Denise from the
there she
Ensembl
is.
team, and I'm going to show you how
I'll
to
close
explore.
this pop-up. But if you're ever not sure, do look out for these question marks because they can help you out. So there are 3 images on this page. At the top we have the chromosome itself. In the middle we have a kind of overview, and then at the bottom we have our detailed view of our region of interest. So the chromosome, this red box indicates our current location in the chromosome. Because we're in human, which is quite an old genome, we have quite old-fashioned data in here. So we have the chromosome banding patterns, which a lot of people still use as kind of sort of geographical signposts as to where they are in the genome. As well as this kind of highlighted red box, you can also see these other red highlights. These are actually haplotypes and patches. If I click on one of them, it gives you this very long, complicated name. They're different versions of the genome that are found at different loci. So this particular locus, it has such different versions between different individuals that it's better to represent it as different versions of the genome than as genetic variants. I'll just close that. We also have the centromere shown here. So if I scroll down a bit more, this bit in the middle is the kind of overview of our region. And again, our red box shows us where we are. In human, the standard size for this is 1 megabase. But if you explore other species, you'll see that the standard size differs, and it depends on the size of the genome and the gene density as to what makes it a sensible size. In this view, you can again see the chromosome band. You can see the contigs, which is shown by these alternating shades of blue. And the genes are shown underneath as these coloured blocks. The labels are shown underneath the genes. So you see this gold block here. If I follow the leftmost point of the gold block down, I find this label FGF2, and I know that that label belongs to that gene. This big gene here, if I follow the leftmost point and that down, it's SPATA5. So if you just follow the thing down, you'll see as I move my mouse around, it's a hand shape and it's got this dotted line. So what I can do, I can use this this view for things like zooming and selecting things of interest. So I could say I want to look at this gene, and I can drag out a box around it, and I can either mark the region or jump to the region. The alternative thing I can do, using this tool up here, where it says drag/select, I've currently got selected this dotted line box, but if I switch to this double-headed arrow, now when I click and drag, it's moving me around kind of Google Maps style. Because this is just the overview, it's not the sort of proper view itself, it asks me in the view at the bottom if I want to update it or reset the this view up here back to where I was. I'm just going to reset back. You'll see underneath the view we've got all of these legends as well. If I scroll down to this more detailed view, you'll see that we have the genes in more detail. We have them broken down by chromosome, and we have the intron-exon structure, which we'll talk about in more detail later. And you'll see that some of the genes are shown up here, and some of the genes are shown down here. And across the middle, we have this blue bar. So the blue bar is the contigs, which I mentioned before is the bits of data that the genome is made up of, the bits of sort of sequencing. And we're currently in this contig AC, blah, 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 blah. And we have these genes that are shown above it. These are actually forward strand, and the genes that are shown below it are reverse strand. So any feature which has strands, so we have things like cDNAs and ESTs aligned to the genome. If we put things on like proteins, they will be shown in this kind of above, below the genome style, where anything that's positive strand is up here, anything that's negative strand is down here. We also have some stuff which doesn't have a strand, so we have these GERP elements. These are highly conserved regions between species. They're not stranded, they're shown very near the top. And we have other non-stranded stuff, so variants, regulatory features. These are also non-stranded, and they are shown at the very bottom. And again, we've got a legend at the bottom of the view. We can change this view so that we can see different things that we might be interested in. So to do this, I use this button here, configure this page, and this opens up a pop-up menu. And this is showing me all of the tracks that are plotted onto the genome. So tracks is a word that we use all the time in genome browsers. It basically means a data set that can be plotted against the genome. So some example tracks we have here are contigs, dbSNP variants, human cDNAs. These are tracks that have been plotted. So we have their names. If I click on this I button, I get a description of what the track is. I have this menu down the side with categories where I can find loads more tracks. So if I go into variation, for example, I could turn on exact short variants as an example. So I'm going to turn that on in normal. So if I know what category my variants are likely to be in, that's the way I can turn them on. If I'm not sure about the categories or I want to find them very quickly, I can also use this find. So if I type in protein, it finds me anything with the word protein in the description and anything with the word protein in the title. So I'm going to turn on this proteins mammal from Uniprot, and I'm going to put that in labels. So there's lots of different styles. Try them out, see what gives you the information you need. Once I've added the tracks I need, I can save and close this menu, which I can do by clicking on this little tick, or indeed anywhere outside the box. and it thinks about it, takes a moment to load. Okay, so it's now loaded with those tracks. So I've got these proteins from UniProt. So we've got their UniProt identifiers. So we've got one at the top, which is aligned to the forward strand. And we've got another one near the bottom, which is aligned to the reverse strand. We also have these exact variants, which I've loaded. And so you can see them appearing there. Now, one of the things I might want to do, if I'm looking at these exact variants, I might be interested to see how they match up to these proteins up here. In the current view, that's not very easy to see, but I can just move them. If I put my mouse over this section on the left, it turns into this double-headed arrow. I can just click and drag, and I can move these all the way up here so they're next to my forward strand proteins, and I can compare them. I can also change the style. I can say actually these proteins are a bit more, they're taking a bit too much space with these labels. So I could click on the proteins, I can go to the cog and maybe change it into stacked, and then it will reload. So now we have the proteins displayed in a much more compact style. Or I could even just get rid of them by clicking on the cross. As with the views higher up, I can click and drag and I can mark this region or jump to it. If I click mark this region, it gives me a highlight, and this highlight remains visible as I zoom in and out until I choose to get rid of it, which I can do by clicking here. So if I found something really interesting in an exon, I could mark it, and then I could zoom out so I could see the whole gene and see the context. I'll just get rid of that marking. Another thing I might want to do, if I found something really interesting and I want to share it with my colleague, I might want to send them a link to this page. Now, if I just select this link up the top here, you'll see it says Ensembl.org, Homo sapiens, location, view. So it's just saying we're on the location page, and then it says what location. It names which database we're looking at, which is not particularly relevant. It doesn't actually mention that I've turned on these exact variants at all. It doesn't provide that information. So if I wanted my colleague to see exactly what I'm seeing on the page with all the manipulations that I've done, I can click on share this page, this link over here on the left. If I've got custom tracks, which I happen to have, it asks me if I want to add them or not. If I do, I can just select accept, and then it gives me a link which I can copy and paste and send to a colleague via email, and they can inspect exactly the same thing that I'm looking at. The last thing I'm going to show you on this page is how to reset the configuration. If you've been doing a lot of messing around with this, rather than having to remember what you put on and what you turned off, you can just use this button here, reset configuration, and that will just take it back to how it was before. I'm using Ensembl to look at genomic regions and find information.